Hey everybody, I just want to go quickly through this lecture here to give you um, material that you can use to study and review for the exam. It's going to be pretty short here and I'll cover only the essential things, okay? So quickly, let's go through the learning objectives here. I want you to be able to recognize this chemical scaffold present in the drug molecules. Specifically here, we're talking about amino acids. Amino acids. Insulin is a peptide and it's made of amino acids. Okay, so I want you to be able to know these amino acids. Compare the activity of insulin analogs based on differences in the structures. Again, here, based on differences in the amino acids. Explain the types of interaction that are responsible for binding of insulin to its target. Understand the chemical structures of these amino acids and also the targets responsible for binding interactions. Predict how modifications, alteration in structure will impact drug activity, potency, and selectivity. And specifically for insulin, we're talking about whether the drug is going to be rapid acting, long acting, or intermediate acting. Identify specific functional groups, specific amino acids that are involved in insulin ADMET properties. We are focusing for insulin on the absorption and the distribution, and explain the mechanisms involved in ADMET using structure-activity relationship. And finally, propose new structures, new drug molecules to improve the current drugs. All right, so let's get started with this lecture here. Now, diabetes is a complex chronic condition and is characterized by an impaired glucose homeostasis with comorbidities that come with the condition. The treatments, you have insulin therapy, non-insulin therapy, and you also have lifestyle changes. Okay. Now, insulin's main role is to facilitate the transport of glucose into the muscle and adipose cells. And it does that by binding to its receptor here and triggering a series of events that's going to ultimately lead to the translocation of the glucose 4 receptor, glucose transporter receptor. Let me say that again. Glucose transporter 4 is going to be translocated from the vesicles to the cell membrane to allow for glucose uptake here. There are many other types of glucose transporters, 1 all the way through 12. Glucose transporter 4 is the one we're going to be focusing on because it is the most abundant and it's the one that's the best studied. All right, taking another look here at the glucose receptor, at the insulin receptor here. The insulin receptor is made up of alpha subunits and beta subunits. The alpha subunit is extracellular, okay, and is responsible for the binding. This is where the binding takes place. The beta subunit is made up of a tyrosine kinase domain. Once insulin binds to the alpha subunit, there is a conformational change inside of the insulin molecule that allows for the beta subunits to get closer together. Okay, insulin binds, the beta subunit get closer together, and then what happens? The tyrosine kinase domain actually begin to phosphorylate each other. So this domain is going to phosphorylate this one, and this one in turn is going to phosphorylate this one. Okay, so it leads to a cascade of events that is going to cause the glucose transporter 4, and let's say that this is the vesicle where the glucose transporter 4 are located, and so this series of events is going to lead to the glucose transporter 4 to be translocated from the vesicle, storage vesicle, to the cell membrane. Once it's transported to the cell membrane, then glucose will be able to get inside of the cell. Now we mentioned how insulin is going to bind to its receptor, and in a little bit we'll talk about specifically about the A chain and the B chain of insulin. For now, I want you to focus on the specific amino acids that are involved in the binding to the receptor, okay? On the A chain, lysine, glutamic acid, and glutamine. On the C terminus, you have tyrosine and asparagine. On the B chain, on the N terminus, you have valine 12, then tyrosine 16, glycine 23, phenylalanine, and tyrosine 26. Okay. Now I've drawn these amino acids here so that you can get familiar with the structure of the amino acids that are involved in the binding. Okay. Tyrosine is here. Phenylalanine is here. Okay. Glycine and valine is here. Right. So I'd like you to be familiar with that, and I'll give you some practice quizzes so that it helps solidify the information in your brain. Now let's talk a little bit about insulin biosynthesis and storage. Insulin biosynthesis and storage. Let me try a blue pen here. Now insulin is synthesized in the beta cells of the pancreas from pre-proinsulin. Okay. Before we go any further, I just want to mention that the alpha cells of the pancreas are also going to synthesize glucagon. So insulin is synthesized by the beta cells, and the alpha cells make glucagon. Right. Pre-proinsulin is the molecule that's going to be cleaved, and pre-proinsulin is going to lose 24 amino acids to generate proinsulin. Proinsulin in turn is going to be cleaved by prohormone convertase 1 and prohormone convertase 2. In this process, we are going to lose the C connecting chain here, and the A and the B chain, because of the presence of cysteine residues. Are going to be connected together by disulfide bonds. So the proinsulin is going to fold on itself, mainly driven by the tendency of these cysteines to form disulfide bonds, and then prohormone convertase 1 and prohormone convertase 2 are going to cleave at this position. Now, prohormone convertase 1 is specifically going to cleave at the arginine-arginine position, and prohormone convertase 2 is going to cleave at the lysine-arginine position here. Now, in patients that are receiving exogenous insulin, one of the ways to determine whether or not the beta cells are still functioning is by measuring the amount of C-peptides that's released. When insulin is released, there is an equimolar amount of C-peptide that's released. Okay, so radioimmunoassay is used as a way to measure the concentration of C-peptide or the amount of C-peptide that's released. And C-peptide amount is a biomarker for beta cell function. Now, what's going to cause insulin to actually be released? There is many different stimuli that will cause insulin to be released. The one we want to focus on today is the presence of glucose. Glucose is going to bind to the glucose transporter on the membrane of the beta cells. Okay, and then this is going to cause insulin to be released by exocytosis. All right. Now, when insulin is stored inside of the granules within the beta cells, it is stored as crystals, and inside of the beta cells you have zinc, okay, and for every 2 zinc atoms you have 1, 2, 3, 4, 5, 6 uh, 6 molecules of insulin. So you are dealing with a hexamer. Now, insulin is going to be released at a high rate when there is a response to a stimuli. Okay, normally there's a basal insulin release. Okay, but in the presence of glucose, you have an increase of that release of insulin. I need to mention that the actual active form of insulin is the monomeric form. It's a monomeric form. So the one that's going to interact with the receptor has to be in the monomeric form. Once insulin is released, it stays in the system about 3 to 5 minutes. About 3 to 5 minutes. It's going to be cleared by the kidney and the liver. So it's very short-lived. It's released, has to get to the receptor. If it doesn't get there and it stays in circulation, it's removed by the kidneys and the liver. Now let's focus on the actual structure of insulin here. Insulin has it's a peptide made up of 51 amino acids, and it has 2 different chains, the A chain and the B chain. The A chain and the B chain are connected together by disulfide bonds. Okay, the A chain has 21 amino acids, and the B chain has 30 amino acids. Now, when you look at the structure of the A chain, you actually find out that the A chain contains an intra-chain disulfide link. Disulfide link. Now, these disulfide links contribute to the stability and integrity of insulin. Again, at low physiological concentration, insulin is going to exist as a monomer. At higher physiological concentration, or even when it's in vials to be dispensed, insulin can dimerize and even form hexamers. All right. So let's talk about the effect of dimerization or hexamers. Now, we mentioned that the active form of insulin is actually the monomer. When dimers and hexamers exist, they are absorbed at a slower rate than monomers. Okay, they're absorbed at a slower rate than monomers. So when we deal with insulin, what we want to do is the modifications that we're looking at are either we make it rapid acting, intermediate, or long acting. And the way we do that is by changes to the chains, A and B chain, or by formulation. And I'll explain that as we keep going. The first analog that we look at is regular insulin. Regular insulin is considered short-acting. It has a 30 to 60 minutes onset of action. So that's the regular insulin, short-acting. Then you have intermediate-acting insulin. Intermediate-active insulin. In this case, we will deal with NPH and Humulin R500. NPH, which stands for neutral protamine hagridon, is made by combining regular insulin with a stoichiometric amount of protamine. And protamine is a positively charged polypeptide. Positively charged polypeptide that's going to be combined with a negatively charged insulin that's going to give us a neutral protamine hagridon insulin. Another way to make intermediate acting insulin is by preparing crystalline zinc solution of insulin in solutions in buffer. And this is similar to how insulin is stored in the cells. Remember, we mentioned that in the cells, the zinc is present and you have hexamers. So in the presence of zinc, at high concentration, insulin can form hexamers, which will slow down the release of insulin and the absorption of insulin. In the case of Humulin R is going to exhibit the profile of an intermediate acting insulin because of its concentration, okay? Mainly because of the concentration. Now let's deal with the rapid acting insulins. The rapid acting insulin are synthesized by recombinant DNA technology, and they're created by making changes mainly at the beginning or the end of the B chain. Okay, the B chain are where most of the changes take place for rapid acting insulin. All right. So those analogs are rapidly absorbed and have a short duration of action. First analog in the rapid acting insulin is insulin lispro. This is an analog in which the proline at position B28 is reversed with the lysine at position B29. Okay, so proline and lysine are reversed, then it gives you the insulin lispro. Now for insulin aspart, there is a single substitution that takes place. Single substitution takes place. It still involves the B28 position, and this time the proline at B28 is going to be replaced here by the aspartic acid. Okay, hence the name insulin aspart. So proline is going to be replaced by the aspartic acid. Still going quickly through those different insulins here. The insulin glulisine, or Epidra, the brand name, it differs from regular insulin that the asparagine at position B3. Asparagine at position B3 has been replaced by lysine, okay? And the lysine at position B29 has been replaced by glutamic acid. Now the three analogs that we just saw are all able to quickly dissociate into monomers and are rapidly absorbed, which is why they have such a rapid action. Now the longer acting insulin are synthesized in such a way that they will self-aggregate in such a way that they are absorbed slower into the body, and so they last longer into the body. They're used for basal type of insulin coverage. The first one that we will look at is insulin glargine. Insulin glargine is an analog that contains two additional arginine molecules attached to the B chain. Okay, they're attached to the B chain carboxyl terminal. So here is the B chain, okay, and here are the two arginines that are attached to the B chain. Now this particular analog is going to be soluble in acidic solution, but it's going to precipitate in more neutral pH of the body. What does that mean? If it precipitates, that means it aggregates. It's not soluble. So that analog is going to be absorbed slower, which gives it a longer duration of action. All right, insulin detamere, also known as levemir. It is synthesized by removing the threonine at position B30 and attaching a 14-carbon fatty acid chain, myristic acid. So let's take a look at this here. 25, 26, 27, 28, lysine 29. So the threonine has been removed at position 30, and instead we have a myristic acid that's been attached. Now that allows, that modification is going to allow self-aggregation to take place. So that increases self-aggregation, and then it's going to lead to reversible binding to albumin. Both the self-aggregation and the reversible binding are going to lead to longer acting insulin. Now, insulin degludec. In this particular analog, we have the threonine at position B30 has been removed, and the lysine at position B29 has been conjugated to hexadecanoic acid via a gamma L glutamyl linker. Okay, so again, there's a theme that's coming back here: the B30 threonine being removed, and then instead of the threonine, you actually have a more of a lipophilic substituent. All right, now because it's formulated with zinc, you have dihexamers that form, but once it's administered subcutaneously, it's actually going to form multi-hexameric complexes, which is going to further decrease the absorption and distribution of the insulin in the body. So this contributes to making this a long-acting insulin. All right. So this picture here is a summary of everything that we've looked at, and it's included here for your reference. Again, regular insulin is here. Glargine has the extra arginines on the B chain, and then the glycine is being added at the 21 position here, and that's for insulin glargine. Insulin lispro, the main changes are we basically the proline and the lysine have been shifted. Insulin glolysine, you have changed at the 3 position where you have a lysine, and at the 29 position glutamic acid is included. Insulin aspart has a single change, and then insulin deramere and insulin degludec, as you remember, we removed the B30 threonine, and then we add the linker, and to that linker we have a lipophilic fatty acid. Alright, this is going to allow for self-aggregation, and longer duration of action. Alright, here are some of the references that I used. I also put references on the Blackboard site to give you some more resources to use if you want more information about this subject. Thank you so much for taking the time to watch and listen to this, and I hope it's helpful for reviewing for the exam. Thank you.